started, and it is my great pleasure to welcome Dr. Becca Lewison to UCLA. We've been trying to make this happen for a few years. So Becca is from New Jersey originally and got her bachelor's at Vassar in biopsychology in 1993. And actually went to Africa while there to the school for field studies, and then started at Davis in '94, where she got her PhD in ecology, studying the behavior and conservation of hippos in 2001, and then she did a postdoc with Larry Crowder at the Duke Marine Lab for a couple of years, and then went to San Diego State in 2006, where she's a full professor in the biology department. So, and we actually met at Davis as graduate students, and actually we figured out literally exactly 20 years ago, like right now, that I was homeless, and Becca took me and my cat in as housemates, despite her policy of not associating socially with any other students. So Becca has many other talents, actually, including a beautiful singing voice, which I've gotten to hear a couple times when I'm 64 at our wedding and at her daughter's bat mitzvah, and doing Lionel Richie, actually, in the Lionel Richie wig, which, for the record, I have a picture of, but I am not showing today. So, but anyway, so I don't know that she's going to sing for us today, but she is going to talk about super cool research she's doing on conservation in all kinds of systems, marine and terrestrial.
Thank you, Seth. No, rest assured, we can safely, there will be no singing today. Thank you so much. I know it seems like I should be coming from a more exotic place, given that it's taken us a while to organize this. I'm just at San Diego State, so if we don't get a chance to talk today, you know where to find me. What I'm going to do, so when people invite me to give a seminar, because my research portfolio is kind of broad, I'm always like, I'm not sure, what is it, which part are you interested in me talking about? And I always have that sort of like, maybe Michelle Wolf now moment of like, is this going to go over the way that, did you invite who you thought you were inviting? But so what I'm going to do, for this seminar, what I'm going to do is organize around this topic of embracing complexity in applied conservation science. So that's where I fit into the ecological universe. I'm an ecologist who focuses on applied conservation science. And as you heard from the intro, and as Seth told you, I work on terrestrial systems, I work on marine systems. Really the unifying theme for the lab is innovating and advancing applied conservation science broadly. And this topic of complexity is something that's come up for me recently, and I've been writing about it and now talking about it. And so I'm going to use that as a thread as I go through these different examples of the research that I'm engaged in. So hopefully there'll be a little bit of something for everyone. I know there's a lot of people with terrestrial focus, but we are going to do a little marine foray. But just so you don't get anxious, where we're going on this is I'm going to talk a little bit about what I see when I'm talking about complexity. What do I mean? Why do we need it? And then these are the examples that I'm going to talk about. So thinking about both seascapes and landscapes, conservation and planning. Then I'm going to switch gears and talk about linking economics and ecology, and then I'll finish up talking about organismal health. So some of these topics may intersect the work that you're doing, but I'm hoping this theme of embracing complexity there'll be something in there for everyone. So I typically, not if this is you, but in general, I can't stand it when people put quotes at the bottom of their emails. Until I read this quote. It's not on the bottom of my email, but if I were going to do one, this would be it. This really resonated with me and sort of was that articulated the struggle that I feel like I typically face in my research. So David White is a poet from the UK, and what I like about this is that it really sort of encapsulates what it is that I want to talk about, which is when we're dealing with applied conservation research and thinking about these complex conservation landscapes, I feel like one of the ways we've been approaching it is to try to sort of change reality by eliminating complexity. So I just need to shout out that I'm not the only person talking about this, about the importance of complexity in conservation and research. So just here are some of the other authors, and this is not an exhaustive list, of other people who are sort of calling for sort of a similar shift in conservation landscapes as a complex system, and even going so far as saying we need a conceptual revolution in conceptual domains. Okay, so when I talk about a complex system, and probably when you talk about it as well, I'm really just talking about a system where we have a high number and diversity of interacting components and elements. And those typically arise because the system is being influenced by multiple processes operating on disparate spatial and temporal scales. And as some of you may know, because maybe this is part of your research or your research program, is that there's an entire family of analyses that get called complex system analyses. Sometimes you'll see this abbreviation, CSA, that are analytical or quantitative approaches that focus specifically on how you deal with that, on linked, or how do you look at a system where there's linked and often reciprocal heterogeneous elements. And so this is, again, not all of them. So when we talk about complex system analysis, it's not like a one thing, right? It's a family of approaches that we adopt when we are looking at those types of systems. And so you could think about this as sort of a continuum of moving from a reductionist, univariate on one end to a complex system on the other. But what got me thinking about this and writing about this was saying, you know, that's not really very satisfying, that sort of one-dimensional way of thinking about complex system analyses. And so in a recent paper, or not recent, it's coming up, an upcoming paper that's actually coming out in the Frontiers of Marine Science, Andrew Johnson and Greg Vrudis and I sort of wanted to unpack that and say, yeah, so there's that distinction of single univariate and traditional complex system analyses, and it's not just like when we say complexity, what are we talking about? So the way we were thinking about it is saying, well, one thing is that there's usually a lot of data. So when you do an agent-based model, if you're doing that, or nonlinear dynamics, you're typically working in a space where you have a lot of data, right? But there's also another dimension of analytical complexity. That's like the one coming out at you, right? But what's happening over here? And what we wanted to call attention to is what's happening over here is something called conceptual complexity. And a term that's being used, it's going to sound like something I made up, and I'll promise you I didn't, is complexity awareness. Sort of this whole parameter space is how even in low data, even with low data availability in data somewhat poor systems, and not necessarily assuming high analytical complexity, how can you bring complexity in? And this term complexity awareness, that sounds like I just made it up, is actually a term that's been used in computing science for like a long time, since the 90s, and more recently is being used by the NGO community, social change community, to kind of, as they're grappling with these complex systems that they're working on. So one of the things we wanted to do was really just highlight, there's no like action items here, but really sort of unpack that idea. When we talk about complexity and embracing complexity, we're not just talking about this, we're not just talking about this. There's an ability of bringing in complexity awareness to research, and specifically, I'm thinking about it from a conservation perspective. So I'm going to bring that with us as I talk about these different examples of the work that I've been doing most recently, and sort of thinking about how that informs the work. Okay, so we're going marine first off, but we'll come back on land in a second. So this work came out of sort of a decade of work that I did, I guess more. As Seth mentioned, all of my training as an undergrad was terrestrial, and then for my postdoc I was at a marine lab. And you may be thinking, how'd that happen? It's an interesting story, but basically the work that I started doing was on bycatch. So just to make sure we all know what bycatch is, when we fish in wild systems, which is like commercial fisheries, not aquaculture, and we use any type of gear. I know it's a net, but I mean any type of gear. Sometimes we catch what we want, that's green. Oftentimes we catch what we don't, that's red. It could be too small, it could be a different species, or it could be a long-lived sea turtle, marine mammal, or seabird. And that's what I spent a lot of time looking at. Like, any question you can think of. How much, where, what gears, what were the spatial patterns? Like, just, basic questions of, for megafauna bycatch, what does it mean and how much is there? And I'm proud of that work, and this was a project, this was one of the culminating projects that I finished up as part of my postdoc, which was to create this map of sort of like, this is what the megafauna bycatch landscape looks like globally. So we're looking at those 3 different taxa, these 3 big gear categories, and bycatch intensity is just a way of saying, like, how bad is it? And that's great. And it was a really useful and piece of information that we were able to compile, and it did, it was pretty substantial. But in going through that process, where that took me was this sense of, these are all interesting questions, but where do we go with this information? Where do we go with this knowledge about bycatch and sort of the bycatch universe? And in particular, led me to this, which is this realization that, like, physical oceanographers know this all the time, right? Biological oceanographers know too. The ocean is dynamic. The species that reside and rely on those oceans are dynamic. And our uses of the ocean are dynamic. Why is it that we then have static management structures that are used to manage dynamic oceans? So when I mean static structures, what do I mean? I mean like fixed time area closures. Like, this area will be closed from April to November. That's like a static structure. And what that led me to sort of this evolution of thinking was saying, oh, we need to be talking about bycatch and actually fisheries management in a different way. And so with a number of colleagues, we sort of led this movement of talking about dynamic ocean management, which was really just highlighting work that was already being done but sort of didn't have a way of talking about it. So dynamic ocean management is just this idea that we can use real-time and near real-time data to support management responses that change in space and time at scales that are relevant for animals and for people. And that's kind of a mouthful, so I'm just going to boil that down a little bit and say it's using diverse data that support management responses. And when I say diverse data, I mean like really diverse data. It can be satellite, remotely sensed oceanographic information, things from a vessel, things from fisheries, genetic information, telemetry, different types of telemetry. So this is not exhaustive, any of these things, right? And so what we're doing then is, if the words don't make sense, here's the visual. For species that move like a lot, even fairly mobile and kind of homebody flounders, if we can reduce the temporal scale of the data that we use to monthly, weekly, and daily timescales, we can reduce the spatial extent of the management actions, right? And so how does this relate to bycatch? Well, it relates to bycatch because we were approaching this problem in a fixed way, in a static way. And we're also approaching this problem as if it were decoupled from the actual activity. The reason bycatch happens is that we are fishing, right? But when we talk about bycatch mitigation, we don't usually talk about it in the context of we want this fishery, if it's sustainable, to continue fishing. So the specific contribution that I've been engaged in and the sort of the cool science that we've been contributing to dynamic ocean management is a project called EcoCast. And I just need to give a shout out to the big research team that's behind this, my partner in crime and co-PI, Elliot Hazen, but lots of other people, our funding agencies, primarily NASA is our major funder for this. And the context for this is basically right off our coast, right? So California, we have a swordfish fishery, and there we are. So what's happened from about the 1980s is a series of closures. Those are these different colors here. The biggest one came in 2001 in response to critically endangered, bycatch of critically endangered Pacific leatherbacks, something we don't want to do. And this big closed area was not like totally random, right? National Marine Fisheries Service put it in the place where we're most likely to encounter leatherbacks. And just not to leave out loggerheads, there's also this extra additional closure that comes in during El Nino events. So very effective, absolutely stopped leatherback bycatch. It also destroyed this fishery. So what we're looking at here is the millions of pounds of fish, and basically this is the series of closures, and you can see, well, you can't see, but we're down to probably about 15 to 12 active permits. The swordfish themselves, it's a sustainable stock, right? We don't have a swordfish problem, we have a bycatch problem. So what our goal, and sort of the idea behind dynamic ocean management, is say, can we hold these two things at the same time? Can we have ecological sustainability? We can't catch, we're legally mandated by the ESA, we can't catch leatherback sea turtles, but do we have to destroy the fishery while we do it? So now we get to the science part. That was just the setup. So the science behind this is basically, on its face, it's just sort of best practices species distribution modeling. The cool thing about this is that it generates dynamic species distribution models. And I'll show you what those look like in a second. So we use telemetry data, here's where that's so important, with historical oceanographic data, create using a statistical model, so there's no process here, this is just pattern, come up with robust statistical fits. We've done a lot of work on what's the best one. We've gone with boosted regression trees, happy to talk about that more if people are interested. And then that gives us a probability of encounter, right? And so part of the cool science that we've been able to do along the way to this very applied science product is to generate these dynamic species distribution models for the target species, as well as 3 different non-targets. There are more, but we've just focused on 3. So if you're interested, you can check those papers out as well. What the EcoCast product is then is an integration of all of those dynamic species distribution models, and what it gives you is a daily map that shows you in this better to, oh, I'm missing my scale, but it's better to fish, worse to fish. So we're basically saying, where do you catch what you want, that's swordfish, and avoid what you don't. So the areas that are red then would have high bycatch and also low target catch. And the reason I'm just showing you between 2012 and 2015, this is a blob year, which means it's warm. You know, there's a blob of warm water off the coast, and it looks different, right? These are dynamic projections. So we create those every single day, and they're on our website so that fishermen and managers can look and say, where is it better to fish, and where is it worse to fish? So that's an example of what dynamic, and this actually is coming out in just a few weeks. Okay. We're leaving the marine realm. We're coming back on land if people feel more comfortable there. And I want to talk about, oh, we're going back. I want to talk about a similar project that I've been working on with my colleague Megan Jennings, who's been leading a lot of this work, that takes a similar approach, not the exact same, but a similar approach to thinking about this in landscape. So one of the nice things about my lab is that I have people in there who have a very marine focus and people in there who have a terrestrial focus. I can't tell you how many times this happens. Someone who's talking about a marine system, maybe it's something about like connectivity, will say, gosh, I'm not sure how to think about this. And someone who has a very terrestrial background will say, all right, well, 10 years ago, this is how the terrestrial folks were thinking about it, maybe this will help. Likewise, in the reverse, it also happens that someone who's been working in marine systems and very used to working in dynamic systems comes to a terrestrial project like this, where we're thinking about ecological networks, and says, wait a minute, are you thinking about how this is going to change, or is your ecological network fixed? Right? And terrestrial systems, people working in terrestrial systems, are later to that game, right? We're just now starting to think about conservation on this changing landscape. So for this project, we're thinking about connectivity and ecological networks and how we think about that in a dynamic terrestrial landscape. And one of the fundamental challenges here, right, is that when we do things on land, they tend to be fairly fixed, right? Here's where the crossing is. Here's where the core area is. We don't think about creating, or we're starting to now, right? But we haven't been thinking about what will this mean under projections of climate change, of land use change, and I'm using fire interval here, but just to sort of recognize, or to represent like changing landscape processes. We talk a lot about fire return interval in San Diego. I guess you probably do too here. Like it's a huge emerging landscape process, right? So how do we think about creating ecological networks, since we know, right, from landscape ecology, the way we have to do this is have core areas and linkages that are connected, right? So we've been thinking of, and you are probably all familiar, right? So when we think about climate change, and there's different climate models that are give us different projections of what those changes are likely to be. Thinking about climate change, a huge one is land use change, right? This is SDSU in 1931. Here's what it looks like today. This was you guys in 1929. Here's what you look like today, right? So land use change is a huge dynamic on the landscape in terrestrial systems as well. And then here's that landscape process idea. So this is the fire return interval, just one of many landscape processes, but also one that is projected to change likely as an emergent feature of both climate and land use change. So San Diego was one of the first ecological networks that was established in the United States, and this is just part of it. And you sometimes hear about it as called, you can't really see this, but the MSCP, but it's just an ecological network, right, that sort of follows that our conceptual or theoretical thinking of how landscapes are maintained, where you have core areas, and they are here, the core areas are just these different colors, and they need to be connected, right? They need to be connected by corridors or linkages or whatever we call them. And so what Megan started doing in our neck of the woods is basically saying, so I know we have an ecological network and it's been established for some time. Is it actually working? And this is actually one of the fundamental questions. We're not really struggling with the science behind developing these, because I think right now, nothing has changed dramatically. We think core areas, connectivity, right? Things need to be able to move. But the ability to say, so we have an ecological network, does it work? Is really hard. So that's the work that she's been engaged in and that we've been working on together, was basically to say, great, we have a network, it's established, we have historical precedent. Does it actually have physical and functional connectivity? And if not, what are the gaps? And another big part of this, of course, in an applied context is like, yeah, what are we gonna do about it? I'm not gonna talk about the what are we gonna do about it, but happy to if anybody's interested afterwards. I'm mostly gonna focus on some of the science we've been doing to support those first two questions. So one of the things that, a little bit, again, sort of thinking about how do we deal with this while we embrace complexity, is that oftentimes when we do connectivity analyses, we focus on a single species. We do that largely because of logistics. It could be because of data, but we really wanted to say, or we really wanted to maybe advance our application on this and say, we're going to try to do this for multiple species. So one of the challenges is picking those species, and this has definitely been a collaborative process, but these are the species that we've picked for this. Of course, when you pick lots of different species, you have lots of different data types, and that means you actually have to analyze them differently. So we have an analytical complexity component of this project as well. So just like, you probably know this, but so when we're thinking about occurrence data, right, we can use that in species distribution models. For telemetry data, right, we need to use different types of analyses, in this case, different types of selection functions. And then some of the data we have is genetic data, which is also telling us something about connectivity. And so for that, we're using landscape genetics. So in and of themselves, this is all stuff other people are doing. I think what's the embracing theme to me on this one is really being able to put these different data types together, the multi-species component, and really be able to integrate the different types of analyses. So I'm not showing you the whole spatial extent just because I don't know, it kind of gets a lot to look at, but this is just part of the output. And so what we've been doing is using those different data sets to look at connectivity, so using things like Omniscape or resistant kernel analyses, again, things that probably if anyone's engaged in connectivity research, those are sort of standard approaches. And what we're doing is then also incorporating land facets. So trying to anchor some of our biological data with physical data of the environment. So that's been something that Paul Byer's team and others have been sort of introducing as we think about using in situ biological data. Can we also bring a little bit more about the physical environment as a way of anchoring some of our connectivity visioning or planning? And so, again, for that little piece of the Earth that we're looking at, what the analyses that Megan's been leading has been doing is kind of telling us, let's look at flow, let's look at connectivity, let's answer that question of, is this network working, by looking at places where connectivity is channeled or impeded or intensified or diffuse, right? Places where it's working really well. So this is one of the ways that we've been trying to help inform that landscape planning and conservation, thinking about that importance of embracing complexity and how we need to bring that. And I should say that from our perspective, this is necessary, but not sufficient. So the part that I'm not telling you is, what do you do with this information? And there, in fact, is a whole decision support tool downstream of this. So I'm not going to talk about it, but happy to, or you can read some of the work that Megan's put out on that. So just to kind of wrap these things up, I think both of these kind of represent this idea of how do we take complex systems, but do it in a management relevant, right, in that applied conservation science context. So to me, the complexity comes from multi-species, multi-objective, right? In the landscape, we're going to have development, but we want to have an intact or functioning ecological network, and that we're thinking about these dynamics. Of course, they are in different time scales, so we don't have a daily map that changes on landscape, but we're thinking about dynamics. We recognize that this is not a fixed landscape, that we have to be thinking about climate change, land use change, and those sort of emergent landscape properties. Okay, so from here I'm going to switch and start talking about another important element that I see in terms of how we embrace complexity within conservation science, and that's extending beyond the ecological domain. This is so old that when I was in graduate school, I was reading about it too. But what's interesting is that we are still struggling to do that within applied conservation science. I'm not saying within the universe, but within this piece of the universe. So I want to talk about the work that I've been engaged in that I think does that and moves beyond just the ecological domain. And I'm just reaching out to two domains here, and it's not that economics is the most important domain, but I think it's a good example of the type of cross-disciplinary work we need to be doing. So I'm going to be talking about transfer effects, and then aligning economic and ecological goals. I guess I'm going to talk about this one first. This one, we're going back to marine, but this one will be more ecumenical. Okay, so this was a paper that we had just come out, I don't know, maybe a month ago. Matt Burgess is the lead author out of UCSB, and this was basically a way of saying, so when we think about bycatch, we don't think about economics, almost never, right? And now that's one field that I kind of feel like I can say that because, right, I've sort of been immersed in that field. There's been very little consideration of the economic impact. And I think EcoCast, the first example I gave you, is sort of a good example of that, right? We did a great job reducing bycatch, and yet, right, the economic cost was tremendous to California fishermen. So Matt was basically herded the cats to say, hey folks, can we align? Is there a way to look at how the economic goals may or may not align with our ecological goals in a broader sense? And that's what this paper is really about. So he wanted to quantify the trade-off. How much would you lose in profits if you reduced bycatch for a particular group of species? So he's comparing estimated changes in the fishing pressure that you'd need to maximize profits. So that's MEY. Any fisheries people? It's like MSY, but a little bit different. That's the shorthand. And changes in bycatch mortality needed to reverse a population decline. So what's interesting here is, of course, on this side of the analysis, we've got almost 5,000 fish stocks, which is like 75% of the global catch we're able to analyze. On this side of the analysis, we've got 20 different species. It's 8 marine mammals and, I don't know if these numbers are going to match up, 8 marine mammals and like 6 turtles and, or maybe more than that, 12 turtles, no, it can't be 12 turtles, something like that equals 20. It's like marine mammals and turtles and seabirds. So you may be asking yourself, like, why are there 5,000 fish stocks here and only 20 here? And the answer is, we don't have a lot of data, right? We only have data on about 20 species to even do this analysis. But again, I think one of the things that I think is so exciting about what Matt did was, even within not data rich, we can embrace complexity. We can bring in complexity if we account for data gaps and uncertainty. So this analysis does that. So just to sort of the short, well, it's a figure in the paper, but so we look at different distributions. We look at what mortality reduction would be needed. So comparing sort of natural mortality to fishing mortality. So how much would you have to reduce mortality to stop the population decline? Identify what fisheries and their relative contribution to mortality, and that's the background. This is sort of the take-home message, which is then what we do is compare, and again, these are distributions because we have uncertainty and we have to account for that and bring it forward. So how do we look at that? How much you'd need to reduce mortality to stop that decline, and what would be the impact on MEY? That's that profit measure. And then, of course, thinking, how much money would you lose? And could you only do this if you can improve targeting? This is like a fishery-specific thing that basically says, if I could target better fish, I could catch the same amount and make more money, or I could actually even catch less and make the same money. So that's what targeting is. It's just sort of like raising the grade of what you catch. So what's interesting about this analysis is that there's, right off the bat there we knew, and we sort of knew this going in, is that for a few species, even stopping all fishing doesn't matter. These are populations that are so low, it doesn't matter. There's nothing, I think we can say that. There may be nothing we can do about these particular populations. But there's some species that would require a big profit loss. Those are the ones where they have the larger colored circles here. But for approximately half of the species we included, the shift from the current conditions, those are the red circles, to less bycatch would not hurt long-term profits. Long-term profits. That's the important caveat of this analysis. It doesn't mean next year you could get as much as you want. It means long-term sustainable profit. So there's some common sense to it, but what's, I think, somewhat, well, why this got picked up, where it got picked up, and why this was a splash paper is because we don't typically think about this applied conservation question in this economic context. And it's essential that we do, right? Because here we're saying, hey, look, they align. Not for everything, it's not going to solve all problems, but there may not be as divergent objectives as we sometimes think, as I sort of put in that teeter-totter picture that I showed you. Okay, so the last part on the economic piece that I wanted to talk about is a paper that we also have coming out in Conservation Letters that's talking about transfer effects. That might not be a word that you're familiar with, but I bet you're going to know what this is, and maybe you've heard a different word used for this. I'll talk about that in a second. The basic idea is this. When we have a shared resource, right, so it's shared because it moves around across an entire ocean, so it's ecologically shared, or maybe it's market shared because we share it across global markets, or it's a common global interest. carbon sequestration, species extinction, biodiversity. When that happens and one jurisdiction and place makes a policy, okay, so I'm in jurisdiction A and I'm very concerned about some type of production that is impacting my environment, resource extraction, whatever it is, right? And I basically say, okay, I'm gonna stop that production. My consumption doesn't change. What happens? What happens is that in jurisdiction B, in someplace else, production goes up, right? I haven't changed consumption, but I can't make it anymore. So what happens? We get it somewhere else, okay? Now, when that, and jurisdiction can be, it's easier to think about it spatial, but it could be sectoral, it could be temporal. There's a lot of different ways of thinking of that shift, but we're just gonna stick with thinking about as a place. So what happens, right? So green means conservation. In this case, brown, sorry, people who love brown, does not mean conservation. And so in jurisdiction A, we improve the conservation of whatever it is we're trying to conserve. Sometimes when we shift it, we don't just shift it, we magnify it. So in this case, not only is B as brown, right, it's browner. So we have moved we've displaced the environmental impact and in some cases made it worse. So if you've never heard the word transfer effect before, if you've studied economics, you've probably heard one of these words. Or if you've studied social science, you've probably heard one of these words. Or if you're an ecologist who does coupled human natural systems, you may have heard this. Essentially, these are all talking about the same thing. So what we were trying to do in this paper is basically say, I don't think we've connected these dots, even though economists know about this, and social scientists know about this, and some ecologists know about this. When we look across all these different sectoral domains of biofuels and carbon and fisheries and forests and agriculture, there are transfer effects. This is a thing, right? We are missing this opportunity to, we're having backfires, basically, of well-intentioned conservation policy because we have not linked the economics behind the ecology, basically. So even though Elinor Ostrom, or whoever you read, like she was my first introduction to socioeconomic and socioecological systems, even though we've been having decades of calls, and it's of course not just economics, but calls that connect social science and natural science, I think there's still some basic disconnects, and this generation of graduate students probably has less than mine, and so maybe this is something we're partially getting at through a broader awareness of the importance and training, but we still need a lot more transdisciplinary progress. And even though I will say that that's happening, again, in the universe, in the applied science universe, it's still really limited. Okay. So my last examples come more to things that like ecologists talk about more, which is organisms. And where as you heard, I'm not talking about hippos, but if anyone ever wants to talk hippos, I'm probably one of the only people you know who can. It's a very small community of researchers. Although there are some people at UCSB, actually, I should say. Doug McCauley's group has been doing that. But I'm not talking about hippos here. I'm going to focus on a marine example and a terrestrial example that I think sort of brings this idea home to organisms and also thinking about how we think about complexity within this context as well. So I'm going to talk about non-targeted screening in some marine things, a small cetacean and a coastal seabird, and then talk about gene expression as a health biomarker in the always awesome desert tortoise. And I just want to give a shout out to some of my current and former students who've been leading a lot of this work. Chris Millo is the seabird lead, Marisa Trago, who's finishing up her PhD, is the small cetacean, and Christina Drake is the desert tortoise. So in essence, like this in fact works even better. Like there's no stretch. Like there could be no more complex system, right, than an organism and its interaction with the environment, so the external environment and how that affects the internal environment, cellular processes, and all the different ways that we measure that. So in some sense, I think we do think of organisms in that way as complex systems. And so one of the things we've been trying to do is sort of bring that forward to the analytical side and think about how our analyses can really mirror that understanding of organisms as complex systems. Here I need to give, so I'm going to talk about non-targeted screening as sort of a first step of getting there, and I want to give a shout out to another critical colleague who's actually, she's an analytical chemist, her name's Dr. Eunha Ho, and she's been pioneering non-targeted screening. So what this non-targeted screening is, and why I think it fits this idea or this sort of thread of complexity, is basically recognizing that when we talk about like, oh, there's something, my organism is exposed to toxins, right? It could be any type of toxins. I'm going to mostly focus on organic halogenated ones, but you may have your personal favorites. But whenever we screen for whatever those are, we typically are looking at a very small list of what that organism is actually exposed to. And Eunha, from an analytical chemical point, when we sort of talked about, oh, we're worried about marine mammals or seabirds, she's like, you're not really thinking about this the right way. Because if you want to know, again, she focuses on organic contaminants, so that's what we have too. There's three parts to this, right? There's what we know about, there's what we don't know, and then there's actually natural organic compounds that we really have no idea how they may interact with the anthropogenic ones. We don't really touch on this, but it's an amazing, really exciting field of research that we're hoping to maybe expand into. But interestingly, even our known, we're not even looking at that because we just look at targeted, right? There's a lot of compounds we know, right? But we don't look for them in the environment or in our organisms. So what Eunha has been helping us do is to basically develop techniques to be able to think about this organic contaminant load without pretending it's really simple, right? Without ignoring the reality of this. I mentioned this is our toxin group that we focus on, halogenated compounds that have chlorines, fluorines, and bromines. So things everybody knows like PCBs and PBDEs, DDTs, these are all halogenated. I know this is not a majestic picture, but it's here to remind me that without this machine, we wouldn't be able to do any of this. So what Eunha has been pioneering is a two-stage gas chromatography. So gas chromatography is not that's not new, but this machine, because it has a two-stage, so it runs it once and it runs it twice using time of flight mass spec, what we're able to do is, so this is the first run, here's the second run, is get this very resolved indication of all the things that are in that sample. So one sample, 10,000 peaks. Which, if you're the person getting that data, you think, holy crap, who thought this was a good idea? But what it's been helping us to do, anybody who does genomics is thinking, yeah, we have that all the time, right? So Eunha has been helping us develop a halogenomic approach of how do we screen all this data, right? And the way we're doing it is using all the known compounds come from NIST. So National Institute of Standards and Technology, who has a library of all of the known compounds. So we use that as a first pass. And then what we're left with is the stuff we don't know. And that becomes a little bit harder. So we are not analytical chemists. How we're trying to sort of move this ball forward is basically saying, well, what if we do some type of clustering analysis like PCA or NMDS and basically say that there's a compound group here that's all anthropogenic. It has 3 unknowns in it. I can't say what they are for sure, but it could be that they are also anthropogenic in nature. So we're getting putative sort of hints of what those things might be, but we are not discovering those compounds in and of itself. But actually, Eunha's lab, that's what they then do, right? As an analytical chemist, she's got the chops to then dig in on this. So we are, in fact, advancing. Hopefully we can use that to support work there as well. So here's an example of some of the data that we can put out then. This is Chris's seabirds and Marissa's marine mammals. What you can see, this one's a little bit clearer that we're looking at all the anthropogenic compounds, the natural compounds, again, which may play a really important role in what happens to the organism itself, and then all these unknowns. And Marissa's been able to take this even farther. So this is telling us relative abundance of these different compounds. Marissa's been able to take this even farther and look at this as a function of sexual maturity. And she's also now linking this exposure, or the exposure to all of this cocktail, and seeing sort of at a functional ecotoxicology way, how does this influence hormones? So we're looking at reproductive hormones, testosterone, so potential like hormone interference or suppression, as well as stress. And where we're taking this, you know, in the next 2 months, because we're looking to get this out, is also looking how this links to gene expression. So we're really trying to say, yes, so we've sort of embraced the complex cocktail, and we really want to try to get to the place of what does this mean, like at the physiological level, what's the impact that this is having on this one's particularly small cetaceans. Okay, but I can't not have a talk. If I'm not going to talk about hippos, I have to talk about this charismatic megafauna. And I want to, again, this is a work that Christina Drake initiated in my lab of using gene expression as a health biomarker. And I know I've been talking to other people in the lab about how other people are using gene expression to link to behavior. So specifically for desert tortoises, one of their fundamental challenges, only one, they have several, is disease transmission. They have this upper respiratory disease that's a mycoplasma, that by the time they show it, like they're super sick, right? And so there's a lot of interest in seeing, can we, as part of applied conservation and protecting this species, come up with better metrics earlier of what individuals may be infected. It also has a huge impact on translocation, right? We translocate these things all the time. We test them using some, I can't say basic, but like ELISA sort of assays. And so I'm going to talk about like how well those are actually representing the status. In this case, we're using gene expression and experimental approaches to kind of untangle that. All right, I'm skipping through this. Okay, so I think everybody knows, right? We're using gene transcription. This is not new. I mean, I know lots of people are using it. It's newer to wildlife, right? Not to people, because people have been using this for like, or not people, people research, right? Have been using this for a long time for like cancer detection. And so we're basically, in this project, we're basically trying to ask, how can this help us inform desert tortoise health assessments? These are some of the genes that, there's 11 genes of interest that have been identified for desert tortoise. Outside of mammal models, like we have so much less information, right? So one of the things that was innovative about this project and the work that Christina's been able to publish has been like, we just don't know very much about how tortoises negotiate and navigate all these processes. The spoiler is it works. It's complicated, but it works. And so Christina's work has been able to show, with desert tortoises, I have to say, there's a lot of captive facilities, and there's an opportunity to engage in work you would not be able to do with anything else. Like I'm going to show you an experiment that she did where you infected individuals, and that's because these were individuals that were going to be euthanized anyway. And so we have the ability to then check our assessment through necropsies, right? And do a lot of things we wouldn't be able to do otherwise. So in one experiment, we were able to look at captive individuals that seemed abnormal physically, and basically say, can we, from a gene expression perspective, detect that abnormality? So it's confirmed through necropsies, and the answer was yes. In another experiment, again, that we were only able to do because these were all individuals that were going to be euthanized, was to be able to say, even when you get fed bad diets, so it's not just disease, we can detect that through gene expression. This is on juvenile desert tortoises. And then directly speaking to that health pathogen issue, if thinking about that upper respiratory disease, as I mentioned, we're able to do an experiment. This is with a colleague at Penn State, where we have a known sick animal that infects naive animals and look at what's the gene expression response and then what's the antibody, that assay response, that ELISA that we typically use. And what was so interesting about this is that we were able to detect, and look and identify gene expression difference at 200 days, although 200 days seems like a very long time. But what's amazing is that from the antibody perspective, it was another 250 days until we picked it up on the antibody response. So clearly gene expression, I guess the take-home message is it's complicated. They're not mammals, not that anybody thought they were, but there is potential here to really use gene expression in a health biomarker way. And I think as we learn more about those genes and sort of how to analyze those data, I think there's a lot of potential there to really innovate and deal with, in this case, sort of the metabolic complexity of these animals. Okay, I'm going to leave it there so we have time for any questions. So I guess I would say, although, at one point when I first read those conceptual revolution, I thought, well, that's a little bit melodramatic. But I guess I'm sort of coming around on this. I do think we need a conceptual revolution in conservation domains. And I think the challenge as we think about applied conservation science is how do we balance data limitation, dealing with uncertainty and data gaps, and embracing the complex reality of the systems that we're working in. So with that, thank you very much for your attention, and happy to take any questions.
There was just an opinion piece put out yesterday or this morning talking about the valuation of nature. I'm wondering how does like kind of stuff, I guess I see from someone who doesn't have, I just read about the shit, I don't do anything with it. How does the stuff you're doing, I see that it would be very important almost for some of these valuation models that we're talking about in terms of what is a deer worth, what is a tree worth, and that kind of thing. So do you see that too? Is that where some of this stuff could go in some of these arguments, or am I just kind of seeing dollar signs and trying to put them together?
Yeah, I mean, particularly if you think about like how we connect the economic domain and the ecological domain, that is one of the ways we've tried to do it, which is through the concepts of sort of ecosystem services. How do we value ecological properties, non-market, and there's an entire field around that. I guess I do think that is one of the domains where we need to embrace complexity. I would argue that probably where we are right now is not enough. But certainly these types of approaches, it can support that. I know that there's a lot of concern if that's the only way that we take into account, right? But I think aside from that, it certainly points to the fertile ground we have and the distance we need to travel to do a better job of that. So I think it relates. Yeah. Do you have a question?
Gene expression as a biomarker. I just wanted to complete the thought in my own mind about why that would be useful. Is it useful because it says something about whether a population has this infection or not? Or is it useful because it makes a prediction about whether the population is going to mount a response to the infection?
I think the answer could be yes. I think the answer is it's telling us information about the physiological interior environment that we don't have any other way. And the question is then, how do we use that? So in the tortoise example, absolutely. The idea is to say it's an early detection. It's like what people are using for cancer, in the human domain. In some of those other examples, like in the contaminant example, I think it's more of just trying to say, yeah, for a long time we've described you have this much of whatever it is. But what we're really interested in, I think everyone would agree, is what does it mean? What's it going to do to you? So I think the answer is yes, it's both of those things.
But in this case, you could test for the pathogen directly, right? It might be more sensitive than...
Well, except that the antibody test that we have is not more sensitive, right? It took in this controlled experiment where we actually infected tortoises, it took 500 days for the antibody test, the best one that we have, to pick up the disease. Gene expression, it still took a really long time. It was 200 days, but it's 250 days earlier. So I would argue that that doesn't mean that's as early as it can get. It means that we still have a long way to go on tortoise gene expression. I mean, just compared to mammals, it's not close. Like the work that out of Keith Miles's lab, when they have like otters that are infected with diseases, like they're picking it up within days and things like that. So, well,
Do you think it could be
yeah.
more diagnostic if you looked at total gene expression?
It could easily be. We just don't have, at this point, the information that we would need. But easily, right? Once there's much more gene sequencing and information for this species, I think the applications will, exactly as you say. I do think we really struggle with the fact that these, like how to deal with animals that are like metabolically dead for a little bit of the year. And we don't think about systems that way, right? We think about mammals and they're gonna express right away, but absolutely, I think that's a good.
So can we vaccinate the tortoises if
There isn't currently, no, unfortunately, from that mycoplasma. It's a bad time to be a tortoise.
So, I have a question.
Yeah.
So are the fishermen like looking daily at those?
Yes.
And taking- Yeah.
So we, it's a total voluntary program, and yeah, I mean at this point this is a fishery that it's in the news, so is really eager to be cleaner, and so they are working to use those predictions. At this point, we don't have like a formal trial. But I think that may come because there are experimental fishing permits where you can use a different gear and fish more. So
But in theory, right, it's not just that they would have less bycatch, but they would also have better.
that's right. And the important thing to realize is that it doesn't mean like, woohoo, we can fish as much as we want. Like there's still a target and a cap and a quota for swordfish. But arguably, if this works, like you could potentially catch what you want sooner. I think what most fishermen would say is, you know what, we knew where the swordfish were already. Like they're very good at their job. But what they didn't know is where the stuff was that they didn't want.
I see.
Because why would you pay attention, right? But they use like, even this fishery, like high tech stuff. So
Yeah.
they would probably say, thanks, we know where the swordfish are. But what's really hooked them on the, haha, no pun intended. What's gotten them excited about this project is thinking about how it helps them fish smarter. And I think that's what really most of the dynamic ocean management applications are really about. So, yes?
It's kind of a legal question, but like, who is the liability if they catch a sea turtle?
It's a good question. This is one of the challenges of working particularly in this fishery because it is so litigious, the environment. Ultimately it's NOAA who's going to get sued, but the thing is they're going to get sued either way. And the bottom line is that it's, of course, these are not like I haven't talked about like all the model validation and sensitivity analyses we've done and what happens if you're missing data and what happens if you have data lags. Like we've done all that, but it is a risk. That's absolutely a risk. One of the things we're trying to do is increase the spatial resolution of this model by using a regional ocean model system, so ROMS model, and integrating that. And so we're hoping that will increase our accuracy, but it's a concern. That is the major concern. I don't know if there's an easy way around that. It's a hard time to be a National Marine Fisheries Service employee, or just that organization they get it from both sides. So, any other questions?
All right, thank you.
Thanks so much.